So the epithelium will be covered when we do the organ, the systems in systemic histology. I'll be showing you the epitheliums. So I'll start with muscle. Now muscle, we have three muscles: skeletal muscle, cardiac muscle and smooth muscle. For the purpose of seeing slides, we usually show you the skeletal muscle and the cardiac muscle. Now look at the skeletal muscle. They are pink eosinophilic fibers, all parallel to each other, and these slides are all focused in 10x. And if you look very carefully, you will see that they are multinucleated and the nuclei are all situated peripherally. On the periphery, where my cursor is, these are just dark blue lines. If you look very carefully, you would be realizing, and they are under the sarcolemma. So we call them hypolemmal multinucleated muscle fibers. And these muscle fibers, actually in high power we can make out the striations. In the low power, striations are not visible. Now look at the muscles, individual muscles, these muscle fibers. In between you can see some loose connective tissue. This is the connective tissue which is in between the muscle fibers, and in the for connective tissue also you can see some dark blue nuclei where my cursor goes, and these are the fibroblasts. The fibroblasts or the, so we have multinucleated cells. So you have to understand that you have to differentiate the nuclei of the muscle from that of a fibrocyte, which is exactly adjacent to the skeletal muscle. Cardiac muscle, again, they are striated muscles, but these striations are only visible in the high power magnification. All slides that we show you for identification or for the histology practical is on low power magnification, and that's enough for one person to identify the slides. Now if you look at these muscle fibers, they are all branching. The skeletal muscle were all parallel bundles of fibers. These are branching fibers, and in the center of these are nuclei, which are nucleated cells, and the nuclei are centrally placed. They are round nucleus which are centrally placed and uninucleated. One muscle fiber has one nucleus, while skeletal muscle showed a multinucleated appearance. And in between here also you can see that there are connective tissue fibers, skeletal muscle, cardiac muscle, and the third type of muscle is smooth muscle. Smooth muscle is, in a way, they are single, they are not long fibers, they are spindle shaped cells, and those will be seen when we see the organ system. We do not show you skeletal smooth muscle individually because they are always associated with the luminal structure, or when we have the uterus or urinary bladder, all those kind of structures we have. Now after muscle, the next tissue that we see is connective tissue, and cartilage comes under a connective tissue part. And this area that you see, this is actually the trachea, but if you can look at this, you see that where just below the epithelium, look at the epithelium. Now since this is a slide of trachea, I'll be just describing this epithelium. It's a pseudostratified epithelium. It's a pseudostratified epithelium. This is connective tissue out here. The connective tissue out here is loose connective tissue, lamina propria. And now let's concentrate on the cartilage. Cartilage is a specialized connective tissue which has lacunae, and in these lacunae lie the chondrocytes. The chondrocytes you will be seeing from the periphery to the center. They are smaller, and as they go to the center, they are larger. They are dividing, and you find them in cell nests. The periphery of a cartilage is called the perichondrium, which has an outer cellular layer and an inner chondrogenic layer. The cartilaginous matrix of a hyaline cartilage is homogeneous, basophilic, and mostly because it has collagen fibers in it. The cell nest which are there, they are dividing, and you can see the around the lacunae there is a dark circular circle which is looking as if it is a capsule, but this is called the territorial matrix. It's actually the matrix looks dark out here, so there is actually no capsule. It's only the matrix which gives this appearance. So you have perichondrium on either side. So perichondrium has two layers, an outer fibrous and an inner chondrogenic layer. Many questions can be asked on cartilage. What are the sides? What is calcification? How is it different from bone? What's the difference between hyaline cartilage, elastic cartilage, and fibrocartilage? You should be able to say this. So hyaline cartilage in fact forms all the compact bone or the long bones that we see in fetal life are always cartilage, and this cartilage's module gets converted into bone. Trachea has hyaline cartilage. As you can see, this is a structure of the trachea. So you have to remember, if you're asked what are the sites, you have to mention the sites. Now, so when we talk about connective tissue, there are three components that you have to remember. What are the components of connective tissue? It's cell, fibers, and matrix. The cells are the ones which are responsible for secreting the fibers, the matrix, and the fibers are secreted. Fibers like collagen fibers, elastic fibers, and reticular fibers are the three kinds of fibers that are found in connective tissue, and the most abundant is collagen fibers, and collagen fiber also has number of types. I'm not going into the details of that because that you have to read when you're doing your preparing for your viva voce. Now this is a slide showing you the elastic cartilage. You can see this is probably a pinna, so because you can see some stratified epithelium on both the sides, and this area, the thin strip that you see in the center is actually the elastic cartilage. Here the matrix differs from the hyaline cartilage. Matrix has more of elastic fibers, so it's a heterogeneous matrix. And if you look at the proportion of chondrocytes in elastic cartilage per unit area is much more than hyaline cartilage. Hyaline cartilage, so that is the difference. The lacuna are also larger in comparison to hyaline cartilage, and the matrix, as I told you, is not homogeneous. It's a heterogeneous matrix. This also has a perichondrium, which has an outer fibrous and an inner chondrogenic layer. But one difference between hyaline cartilage and elastic cartilage is hyaline cartilage can ossify and get converted to bone. Elastic cartilage never ossifies. And where does we find our elastic cartilage? The pinna, the epiglottis. You have to learn the sites where one has elastic cartilage. You can be asked this question. And when we see fibrocartilage, I don't have a slide, but it's similar to a tendon with some isogenous groups of chondrocytes present in the intervertebral disc. You have fibrocartilage, or where a tendon gets inserted into the bone, one has fibrocartilage. Now compact bone. Compact bone is the part of a bone which has the Haversian system. Now bone itself is also is a specialized connective tissue. That means it has cells, it has fibers, and it has a matrix. The matrix out here is inorganic matrix along with so it's a calcified matrix. It has the calcium and phosphorus salts are there. So it gives it the rigidity. Another difference between bone and cartilage is avascular, gets its nutrition by diffusion, while bone is an extremely vascular tissue. So in this rigid compact bone, which we see, the osteocytes are arranged in a lamellar fashion, which is called the lamellar arrangement or the Haversian system, and it forms an osteon with a central canal, which is called the Haversian canal, and in this Haversian canal traverses the Haversian vessels and lymphatics. This system, if you see, the concentric lamina have osteocytes around it. The osteocytes are lying in the canaliculi, and these osteocytes have minute processes which are making connection with the adjacent lamina, and this is the way nutrition traverses from the central canal to each and every osteocyte. So the osteocytes are lying in these thin canals which are surrounding it, and it gets its nutrition. Therefore, if there is a fracture, there is a lot of bleeding from the bone, the side, and there is a lot of fluid loss. Haversian canal, when they connect with each other, you can see my cursor going. This is called a Volkmann canal. So the arrangement of a cortical bone of a long bone is compact because of the arrangement. The growth of bone, you can be asked, how does bone grow? What are the methods by bone growth? Because it's a tissue which keeps on remodeling throughout life. Has the calcium pool of the body. If there's requirement, calcium can be released into the bloodstream. The cells of the bone are osteocyte, osteoblasts, and osteoclast. The bone forming cells are the osteoblast and osteocyte, and the cells which are which cause resorption are the osteoclast, which are usually on near the periosteum. We don't have a periosteum in this section that I am showing, but periosteum is on the outer surface. Like cartilage, you had perichondrium. Here there is periosteum, which has an outer fibrous and an inner cellular layer. You have to learn about ossification, what is membranous ossification and what is cartilaginous ossification, or we say endochondral ossification. You can be asked questions on that. This is a cancellous bone. A cancellous bone has thin spicules and trabeculae. And here also the bones, if you see, they are the osteocytes present in the lacunae, and they also form a lamellar arrangement. In between the trabeculae, there is marrow space which has fat and hematopoietic tissue. You have to understand where are the sites where you have cancellous bone, which are the sites you have compact bone. The compact bone slide that you saw, is an unstained slide. It's a ground section. That means it has been prepared by grinding the bone into thin slices and just kept under the microscope. It has not been stained at all. While a cancellous bone, yes, they can be stained, or a compact bone can be stained after, or we can process the cancellous bone after decalcifying it. Then one can process a compact bone. It becomes soft to process it. Now comes the vessels. Muscular artery. Any vessel that we see has 3 layers, tunica from, and whenever you are answering or you are doing the diagrams, preparing the diagrams, you have to do it from inside out or from outside inside. It has to be in order. So we have tunica intima, tunica media, and tunica adventitia. Tunica media intima has endothelium, very thin subendothelial tissue, and then you have an internal elastic lamina. The internal elastic lamina is very prominent out here because the endothelial layer is not visible. So after the internal elastic lamina is the media. The media is mainly made up of muscle, smooth muscle cells. So one has to understand that the media has lot of smooth muscle cells, and tunica adventitia is again a connective tissue, a loose connective tissue which is encircling the blood vessel, and through that there are a number of nerves and vessels which are present, which are called the vasovasorum. In this slide probably we are not able to see, but here in this corner probably there are some vasovasorum. Unfortunately we have not been able to give you slides this year because of COVID, so you have not been able to actually see slides and scan them on your own. Hopefully when you go to pathology you will be able to scan slides on your own. So this is a large vessel which is the elastic artery. So elastic artery has tunica intima, media, and an outer adventitia. If one looks at this vessel, it's a very large vessel. Like muscular artery, you could see the entire lumen fitting into the single frame. While elastic artery, it's only a circumference which is fitting into a single frame. That means the size of the elastic artery is very big. It's a large luminal structure which again has an endothelial layer. It has an subendothelial connective tissue. The internal elastic lamina out here is not very prominent because they are all tunica media is made up of lot of elastic fibers with smooth muscles interspersed with them. That's why it's not that it's not present, it's present but not very prominent as compared to muscular artery, because of the tunica media of muscular artery has more muscles. Here there are more elastic fibers, and the tunica adventitia is on the external side where you will also see some vasovasorum. So the nutrition of the blood vessel comes from the blood vessels of the vasovasorum which are coming and supplying the outer part of the blood vessel, while the inner part of the blood vessel is by the blood that these vessels are carrying. So the blood that they are carrying gives some amount of nutrition to the inner 1/3, while outer 2/3 are getting it from the vasovasorum. And then there is veins, and when you look at the veins, they are very large structures, large veins, but the luminal in large veins, the wall will be much thinner and they do not have a regular lumen like the artery. It's a convoluted lumen, right? So in some slides, there might be a vein which will ask you to identify. So you should be able to differentiate between an artery and a vein. This is thick skin. Thick skin has stratified squamous epithelium with a keratinized thick layer of keratin. This is the dermis. You should know the layers of the thick skin, epidermis. What are the layers of the dermis? You should be knowing that this is a papillary layer and the reticular layer, that you should be able to say what are this. And in thick skin, there are no hair follicles and sebaceous glands, they are only sweat glands. So in this section, of course, we can't see any sweat glands, but you should be knowing what are the sides of thick skin, knowing what are the sides, what are the layers of the epidermis. I'm not going into that detail. Similarly, in thin skin, again, it's a stratified squamous epithelium, right? What are the layers of this? You should be able to say. And here you can see a cut section of a hair follicle. This is the root of the hair. This is the shaft of the hair. And sometimes you might be able to see the hair come projecting out, but in this section we can't see. And look at the keratin layer. The keratin out here is a thin layer in comparison to the keratin which we saw in the stratified squamous. This whole expanse that you see is the keratin. So it's a stratified squamous keratinized epithelium, a very thick keratin. While this also is a stratified squamous keratinized epithelium, the keratin layer is much less. Out here you can see some tubular structures. These are a part of the sweat glands. So this is a sweat gland, and here there is a small circular thing. Probably it's a part of the sebaceous gland, right? So this is the trachea, which I showed you this slide before. So we have pseudostratified epithelium. Look at the pseudostratified epithelium. Look at the connective tissue out here. Some elastic fibers. There'll be some sections might show you some sweat glands and some serous and mucous acini. Can't see them well here. And we have the hyaline cartilage, the thick expanse of hyaline cartilage out here. And what is pseudostratified epithelium? How is it different from a stratified epithelium? You have to know all these features. The lung, what are the parts of the lung? What are these structures? You have to know the respiratory tree very well. Now you can see this tubular structure. It's lined by, the epithelium is not very clear, but it would be a pseudostratified epithelium. Look at this part out here. It's lined by a thin plate of cartilage. So once it's lined by cartilage, we call it an intrapulmonary bronchus. The trachea, the bronchus, and the intrapulmonary bronchus, like the extrapulmonary bronchus and the intrapulmonary bronchus, all have a cartilage plate lying surrounding them, and the rest of the parts have muscle cells which are surrounding them. You have to understand this. And if you see the other structure out here, this doesn't have any cartilaginous plate. So this is an bronchiole. So you have to know how to differentiate between a bronchiole, between an intrapulmonary bronchus, between the alveoli, which are just small air spaces lined by simple squamous epithelium. What are you have to actually trace the respiratory tree from the trachea, the bronchi, intrapulmonary bronchus, the divisions of the bronchus, and then going into the bronchioles, the tertiary bronchiole, the respiratory bronchiole, the atrium where they open up, and then the alveoli. Along with them, you will be seeing some blood vessels. In this section, we cannot see any blood vessels, but yes, at places there are some blood vessels also. So that also you should be seeing, right? So that way you can identify the lungs, one of the easy slides to identify and draw. So whatever sections I'm showing you, these are from this only we will be giving you practical exams. Please learn how to draw the diagrams. You can practice them. You've got enough time now to practice your diagrams, and use only HNE pencils when you are drawing your diagrams. So let's look at the salivary gland. The salivary gland is, they have, if it's a sublingual, it's only a mucus gland like this from the tongue. So only mucus glands. But if it is a mixed salivary gland, you will be having both serous and mucous acini. So serous and mucous acini, you should be knowing the difference between serous and mucous acini. You can be asked to draw, but here anyway not showing one which has both serous and mucous acini, but we can show you. So you should know the difference. The tongue, the dorsum of the tongue has stratified squamous epithelium, and these are the projection that it has. These are the papillae. So the thin pointed papilla are the filiform papilla. They are there on the dorsum of the tongue, which make the tongue surface rough. You have a lot of muscle cells. The body of the tongue is made of muscles. You all know about the intrinsic and extrinsic muscles which are present in the tongue. And on the ventral surface, there are some glands, the sublingual glands, as you can see here. So these are all mucous acini. So this is the surface of the tongue. You can see a stratified squamous epithelium on the surface. Here you can see a very thin layer of keratin, but usually it's a non-keratinized epithelium. This is the muscle, and these are the glands out here. The taste buds are present in the circumvallate papilla and the fungiform papilla. The filiform have no taste buds, and it's usually on the side of these circumvallate papilla. If you look at the structure, they are big, having a big expanse on the superior side, and inferiorly it is narrow. The taste buds are on the lateral side of this circumvallate papilla, and circumvallate papilla are usually near the sulcus terminalis. So you have to see that where they are present, and you have to identify these are the taste buds. This is another papilla, the esophagus. Now when we come to the GI tract, starting from the esophagus to the rectum and anal canal, they have 4 layers: the mucus, the submucosa, the mucosa, the submucosa, the muscular externa, and the adventitia or serosa. So 4 layers are common for all GI tract slides. So in that, we have to first identify the mucosa, which has again an epithelial layer, a lamina propria, and a muscularis mucosa. Now if you look at the epithelium is a stratified squamous epithelium. A thick epithelium which is present in the tongue, in the oral cavity, in the esophagus, right? The muscularis mucosa is not very prominent out here. The lamina propria is also thin. There is submucosa out here, and in the submucosa and parts, the upper part and the lower part, upper 1/3 and lower 1/3, have glands. The middle 1/3 has no glands. And then we have muscularis externa, which is arranged in inner circular and outer longitudinal. So inner circular and outer longitudinal, that is the way they are arranged. And outside we have the adventitia layer. So we look at this slide. This is again a stratified squamous. It's one portion of the esophagus. That was a rat esophagus. This is a human esophagus. So you should be very careful while identifying this. Look at this. It can confuse between the tongue, because that also has stratified squamous epithelium. But here, what are you noting? You are seeing that there are no papillae. There are no papillae, just the stratified squamous epithelium. Then you are having a lot of submucosa, and in that you are having some glands. That's all which is visible out here. So you have to identify it accordingly. Look at this tissue. This is the stomach. The stomach, from the esophagus starts the stomach, and the epithelium immediately changes abruptly from the stratified squamous to columnar epithelium. So it's simple columnar epithelium which is lining the entire stomach, And if you look at the stomach, there are, because this is in a very low magnification, they dip into, the epithelium dips into the tissue, and these are called the gastric pits. The entire lamina propria, which is this expanse that you see, you can see this layer of muscle. This is called the muscularis mucosa, and this entire expanse is the lamina propria. In the lamina propria, you can see that the presence of the gastric gland is a difference which is seen, or a characteristic feature of the stomach fundus. The body of the stomach and the fundus of the stomach have the same feature, and if we just try to increase the magnification, these gastric glands are lined by cells, and these large pink cells that you see, it looks as if it's a strand of pearls, or we say it's like a strand of beads, with red color beads, which are the bigger ones. These are the parietal cells. So you have to know what are the cells which are lining the gastric glands. It's mostly mucous cells, the chief cells, and the parietal cells. So you have to know them, what are the cells present, and the gastric pits. Into one gastric pit, 2 to 3 gastric glands open up. So this is a pit where we have 2 to 3 gastric glands will be opening up, the muscularis mucosa, the submucosa, and then one has the muscularis externa. So this is the pyloric part. Pyloric part is the pits are larger in comparison to the body and fundus, and the cells out here, the gland, are mostly mucous cells. There is no parietal cell in the pyloric part, because the parietal cells are the ones which are secreting HCl. So you should know what are the cells lining them and what are their secretions. You have to learn them. So here we don't have any parietal cells. Occasionally one might have, but otherwise there are no parietal cells. Chief cells, it's mostly mucus secreting cells, and they are extremely coiled, because if you remember the pyloric region, it's a narrower part in comparison to the body of the stomach. So the glands out here also get folded, so they become a big coiled glands that are seen in the pyloric part of the stomach. And then you have the submucosa and mucosa. Then coming to the small intestine, duodenum, jejunum, and ileum. Here the characteristic feature is the epithelium is thrown into folds. You have to remember that all the time the structure is correlating the function of that part of the body. So the structure of the stomach was a large bag which was a reservoir for food, and the digestion process sets in. The proteins have to be broken down, so the acid out there is secreted by the parietal cells. When it comes into the small intestine, absorption and digestion starts. So the rest of the food particles, which are the amino acids and the fats, will be getting absorbed and being broken down into their individual components, into the chylomicrons and amino acids, and the proteins, and the carbohydrates being broken down into the glucose. So absorption would be taking place. So for absorption, the epithelium is thrown into, or we say the mucosa is thrown into folds which are called villi. They are thrown into folds which are called villi. So throughout the small intestine, the main characteristic feature is that they are thrown into villi. The shape of the villi, of course, can differ, but the thing is that we just remember that they are thrown into villi. And what are villi? They are surface projections of the mucosa. So what is the mucosa? It is epithelium, it's the lamina propria, and it is the muscularis mucosa. And in these villi, one would see lacteals, which are the lymphatic channels, And these villi, if you bring them down into the submucosa, in the submucosa, you will find that there will be the intestinal glands, which we call as the glands of lubricant. So there are a number of intestinal glands which are present in all of this. The basis of these villi will be having the crypts of lubricant. But in the duodenum, what do we see? We see in the duodenum, we see number of submucosal glands which are mucosal secreting, which are there to nullify the acid of the stomach. So the Brunner glands are present in the duodenum, while jejunum has no Brunner glands. The submucosa has no Brunner glands. It's only connective tissue with inner circular and outer longitudinal layer in all three layers. So the characteristic feature of duodenum is Brunner's gland. In the jejunum, there are no Brunner's gland, while in the ileum, you find that there are lymphatic nodes throughout. There are lymphatic nodes throughout, and the villi have now become slightly blunter. These villi that we see out here, they are all lined by epithelium with microvilli, because it increases the absorption of the food particles, and it also helps in absorption the microvilli which are present. These are the pair patches, which are lymphatic aggregations. Confluent lymphatic aggregations present in the submucosa are called pair patches. Just in front of them, this part of the villi, they will be in between, they will be the microfold cells, or we call the M cells. Now let's see individually. Now look at this. This is the section that we have out here. These are the Brunner's glands in the submucosa. So many mucus secreting glands in the submucosa, and the villi. You can see the villi out here, the connective tissue, and then again the muscle layer. Similarly, the jejunum, you look at these thin finger-like process. These glands that you see are the crypts of lubricant on either side. Then there are the muscles. This is because of the fold of the intestine. We are seeing it from both the sides. Ileum, again, if you look at the ileum, these are the lymphatics. And this is actually, there is, during processing, there's some tissue space has been formed. So you're finding this gap, but just concentrate on these thin villi out here. And these aggregations that one can see out here, these are the lymphatic nodules. Now coming to the large intestine now. After the bolus comes to the large intestine, all the fluid that is present has to get absorbed. So there is no villi. There is no villi in the large intestine, and you see instead the crypts of lubricant, and these glands are all mucus secreting glands. They are all mucus secreting glands, the submucosa and the muscularis externa. Of course, you all know about the muscularis externa in the colon. How is it different? You know about the tinea, so I'm not going into the detail of that. And from there, you have to understand the difference. So these are all the mucus secreting cells which are present out there. Similarly, in the appendix, small luminal structure with lot of lymphatics. Structure is almost similar to the colon, but it has a very thin lumen, right? So from that you can differentiate. Now coming to the liver, central vein, and on the periphery, and look at from the central vein, you see the portal hepatocytes moving out in a radiating manner. They'll be moving out. So the typical picture of a liver is it's hexagonal with the hepatocytes coming out, and at the edges of these would be the portal triads, which are having the portal vein, hepatic artery, and the bile duct. So you have to understand what are sinusoidal spaces, how the blood comes into the liver, how it goes out of the liver, what are the bile, how does the bile flow, which is centripetal, which is centrifugal, like the blood from the portal triad are going towards the center, so they have a centripetal direction, while the bile is coming from these cells and going towards the periphery, so it has a centrifugal direction. What is a liver lobe, classical hepatic lobe, what is a portal lobule, and what is a liver acinus. You have to do that because the most important functional classification is on the liver acinus, and these are the parts which you have to study. I'm not showing you here because it's just a revision on identifying the slides. So the pancreas, again, a part of the GI system which has both exocrine part and endocrine part. So exocrine part is all the serous acini, and when we look at the serous acini carefully, you'll find that the pancreas is a lobulated structure, so it's all divided into lobes by these connective tissue septa, and you can see that in places out here there are no serous acini, but they are clusters of cells which are paler staining, and these are the ones which we call as the islets of Langerhans, which are the endocrine part, which secrete which have the alpha cells, beta cells, and the delta cells, which are secreting insulin, glucagon, and somatostatin. So you have to understand periphery. We have the beta cells centrally, but of course in an HNE section it's difficult to say which are the alpha or the beta cells, but you can identify because of the pale staining in comparison to the rest of the parenchyma of the pancreas. You can make out which is the islet of Langerhans and see the blood flow. So when you're asked about the secretions, you have to answer well. This can confuse between a serous secreting gland, which is the parotid gland. So when we look at the parotid, which is completely serous acini, and this has the islet. So it's with the connective tissue because that is invested with the deep cervical fascia. So there is a lot of connective tissue there in comparison to this, and the ducts also out here, they are the tuboacinar ducts, but unfortunately I don't have an high part, and it's not a class where I can show you what is a centroacinar cell, but you can be asked the difference between parotid and pancreas, so please revise that. So pancreas is also considered a part of an endocrine gland, so we'll do the endocrines. Next is the suprarenal or the adrenal gland, which has both an adrenal cortex, and the central part is the medulla, so it has a capsule. The first layer out is the zona glomerulosa, then fasciculator, and the third layer or the innermost layer is reticularis. Fasciculator as because of the layer wise, the longitudinally placed cells that we call them fasciculator. In glomerulosa, there are like the kidney glomeruli, round arrangement of the cells, so it's called glomerulosa. And in the third part, it's the reticularis, where they are arranged in a reticular manner. And the center part is the medulla, where the cells are also lighter staining, and they are also like the reticular layer. Each layer you have to understand what are the hormones being secreted by zona glomerulosa, fasciculator, and reticularis. You have to learn and what are the secretions of the adrenal medulla, the epinephrine and non-epinephrine. Glomerulosa, like we have the mineralocorticoids, we have the zona glomerulosa, the fasciculator, the cortisols, glucocorticoids, and then we have the sex corticoids from the reticularis. You have to learn them, what are the secretions of each and every part of the adrenal gland. So it can be, sometimes we call it the suprarenal gland, and the which are synonyms. The thyroid. And one thing you have to understand, that each of these endocrine glands are always associated with number of blood vessels or sinusoidal spaces and capillaries. So these blank spaces that you are seeing, they are actually all capillaries, because the secretions are all poured down into the blood. So this is thyroid gland, another endocrine gland which is secreting thyroid, lined by, these are the follicles. In between you have the inside, these are the thyroglobulin. The thyroglobulin, this is not the thyroid hormone, but you will be asked the secretion of thyroid, you can be asked, and how does it get secreted. Look at the lining epithelium, these are cuboidal epithelium, right? And in between we have lot of connective tissue stroma, right? I hope all of you are with me. Yes or no? I can't actually find out. So how many? There are 161 students present for this class, and let me ask somebody, are they there with me or not? Are there any questions? Okay, right, let's finish and then we can have, if there are any questions that you want to ask. So the pituitary, if you see the pituitary, it has anterior pituitary and a posterior pituitary. Anterior pituitary is the cellular part, which has lots of cells, which are called the chromophils or chromophobes. The chromophils are again the acidophils and the basophils, and the chromophobes are the ones which do not take stain. And in on the posterior side, we have the pars nervosa. So the pars anterior, pars posterior, in between is the pars intermedia, which sometimes we see a lot of colloid out here. And the posterior pituitary, in fact, gives an appearance of the nervous tissue. So in high part, we can see the cells of the chromophil, chromophobe very well with the intermediate blood vessels. But here in the posterior pituitary, we just see some fibers, and the terminal part of these fibers are called the herring bodies, and in between we have pituocytes. I've not labeled anything because the slides that you will be getting will be like this only, and from that you have to identify. So when you see one part cellular, one part lighter, you will know it's the pituitary. This is an easier slide to identify because of the kidney, because of the glomeruli. So the kidney cortex, which has the glomerulus, it has PCTs and DCTs. You can be asked what are the luminal structures which are present in the kidney. So you have to answer about the PCT, the DCT, the collecting ducts, what are the lining epitheliums of each part, what is the difference between the kidney cortex and the kidney medulla. You have to write down each and every identifying feature. You can be asked, so you have to understand and read all that. This is an easy slide to in fact identify. There should be no mistake in this slide. Those who get this slide should never do any mistake, because it's one of the easy ones to identify. This, lungs, bone, they're easy. I do understand pituitary can be difficult at times to identify, but yes, you get chances to do it. And ureter, again, if you look at, it's a luminal structure with an epithelium which is a transitional epithelium. So you should know what the difference between all these epitheliums, the pseudostratified, the stratified squamous epithelium, and the transitional epithelium. And look at the layers. This is star-shaped lumen, and the lamina propria, we have muscle layers out here, 3 layers of muscle layer. So inner longitudinal, middle circular, and again outer longitudinal, slightly opposite than that of the GI tract. Urinary bladder, again, lot of muscle. The epithelium is a transitional epithelium, and with that you can see the thick muscle layer. So this is the urinary bladder. Now this slide that you see is the lymph node. It has capsules, and just subcapsular you have these lymphatic nodes. So this is the part of the cortex. The medulla would be having the medullary cords. So when we ask you about the lymph node, you should be able to say what are the lymphatic nodules, what is the germinal center, how the lymph flows from the cortex to the medulla, and what is the function of a lymph node. You should be able to say that. This is showing you the capsule, a thick capsule, and showing you the lymphatic flow. These are the lymphatic channels. Then the spleen, the parenchyma of the spleen, which is made up of red pulp and white pulp. So white pulp is where we have the lymphatic nodules. The red pulp is all the sinusoidal spaces with the RBCs. And the white pulp has an arteriole with lymphatics around it. So that is the identifying feature. Lymphatic nodules with an eccentric arteriole. Here also you can be asked about splenic circulation. What are the types of splenic circulation? You have to understand and explain those to the teacher. So this is also there. These are the lymphatic nodules of the spleen. You have to, with arteriole, if you look at this is the eccentric arteriole. So you have to see them and identify and answer. And the other lymphatic organ that one sees is the thymus. And if we look at the thymus, it's an extremely convoluted lobulated structure which has no lymphatic nodules. The lymphocytes are dispersed in such a way that the outer side is darker and the inner part of the lymph node of the thymus is lighter. And this is called the medulla. The medulla is confluent while the cortex is separated by the capsule and incomplete septas. Incomplete septas are making it in such a way that the medulla of each lobule becomes confluent, and in the medulla you find the Hazel's corpus, like you see these. So the parenchyma of the thymus is made of epithelial reticular tissue, while the spleen also has a capsule with trabeculae. The epithelium, the parenchyma of lymph node and spleen and palatine tonsil are made up of reticular fibers which do not stain with hematoxylin. Central nervous system, cerebellum, outer layer, it has 3 layers. Like the cerebral cortex has 6 layers, this has cerebellum has 3 layers. So the outer layer is a paler layer called the molecular layer, then a single layer of cells which are the Purkinje cell layer, and then you have an inner, or the third layer is a granular layer. In between the granular layer, then you have the white matter of the cerebellum. So you have to identify the layers. What are the cell layers? What are the cells present in the molecular layer? What are the cells present in the glandular layer? And the neural circuit of the cerebellum, you should be able to explain to the teachers. How does the Purkinje cell send its impulses? What kind of impulses are sent and where do they go? You have to be able to say that. Then the spinal cord. Look at the spinal cord. This is only a hemisection of the spinal cord. If you look at it, there is a paler area and there's a darker area. The pale area is the white matter, the dark area is the gray matter. Now let's look very carefully at the dark layer. You can see this is almost reaching the periphery of the spinal cord. So this is the dorsal horn. This part is not reaching the periphery, so this is the ventral horn. So this is the ventral horn and that's the dorsal horn. And in the ventral horn, one has these cells which are the multipolar neurons, right? So from that you can see the multipolar neurons, how they are. So this is at low power. And of course in the central is the central canal. So this is the anterior median. The posterior median fissure, you have the anterior median sulcus. So the ventral horn is not reaching to the periphery. From that you can make out. Of course, it's very light, very faint. I don't know how you people will be seeing, how is it visible on your screens, because it's paler. And of course, I'm not labeling them. I have not labeled them because it's revision. You have to identify it like this. This is testis. Again, tubular structures. These tubular structures are the seminiferous tubules. You have to know what are the cells lining the seminiferous tubules, what's present in the lumen of these, the spermatids, how do the sperms mature, what is it, and these are the lining cell. Epithelium basement membrane, along with that the muscle cells are the myoid cells. In between the tubules you have some interstitial cells which are the Leydig cells. You have to know all the cells, the steps of spermatogenesis to actually say the spermatogonia, the primary spermatocyte, secondary spermatocyte, spermatids, and then how does the sperm mature. You can be asked any question from this. Easy slide to identify. Again, another slide, easy slide to identify. Small lumen with a very thick wall, a very thick wall. In the center, this is the spermatids. These are the vast difference, where you have a very thick muscle coat which is surrounding it, and it's a pseudostratified epithelium, right? Because it has to propel the spermatozoa to move upwards. That's the reason why it has a very thick muscle coat. Prostate, again, the prostatic is present at the prostatic, near the prostatic urethra, on either side of the prostatic urethra. It's a fibromuscular glandular tissue. So you find muscles in there, you find fibers and the glandular tissue. So this can be sometimes confused with breast, but anyway, we are not showing you breast, but because that also has a lot of fibers, but it has a lot of fatty tissue. And if the prostate is a prostate of an old person, you have a lot of amyloid deposits out here, but these have just the glandular part you can see, and some fibrous part and few muscles. The female reproductive, the ovary, you have to understand. When you see these follicles, you know it's the ovary. You have to know all the stages of the follicular maturation, starting from primordial follicle to the Graafian follicle. When does it rupture and what are the parts? Of course, we can't see any corpus luteum in this section, but we can see a lot of primordial follicles, and we see a antral cavity and a maturing oocyte. And what's present in the cortex? The fallopian tube, again, it's pseudostratified epithelium. You see this, the folds which are present here. They have primary folds, secondary folds, and tertiary folds. The whole lumen is just filled up with the folds of the uterine tube. Again, a pseudostratified, the ciliated epithelium, ciliated columnar epithelium, which helps in propelling the ovum towards the uterus. So one has to